Easy champions, I believe every one of you who is watching this video is doing absolutely good. So Baba, first thing before I go ahead, my name is Jasmine for those who do not know me. Second thing is that if you are someone who are not confident enough about the chapters that are there coming for your FA1, okay, please note, and that's my promise, I am going to make sure each and every doubt which is there, it is going to be cleared. You are going to be pretty confident by the end of this particular week. I'm coming with 2 marathon sessions that is definitely going to help you out, and all the questions that I'm going to show you over here are going to be discussed there such that you do not have any confusion. Okay, works. I'll show you the dates and all in the coming time. Let's go ahead, and I do not want to waste your time. So let's see all the 2 markers which are important over here. And what are this? First one, draw a label diagram of the mature 2-celled pollen grain. And I want to tell you one more very important thing, that is, when I say that the diagram based question can be coming, draw a label diagram of mature 2-celled pollen grain, I am also giving you a hint that if at all you have very well understood the diagram of the pollen grain, you will be able to write about the pollen grain as well. When you learn the diagram of the pollen grain, you know the outer layer is known as exine. It is having pores, and basically that pores is because if there is no spore or pollen. Please make sure, if you are someone who is more inclined to do ratification, don't do that. While I say that this question is going to come, it's not like the exact question could come. There could be some changes, and no one in this world can come unless and until it's a person who made the FA1 paper. They cannot come and tell that this question will be exactly coming. Do note, if I say that this question is coming, there could be something coming around the pollen grain. And when you draw a label diagram of this particular pollen grain, you get to understand all the other features as well. That is the expectation a teacher would be having. Okay, so make sure you know the concept really well. Whenever I show you something over here, concept should be clear. Okay, the concept behind it should be super clear, then only you will be able to answer any question that comes from that particular topic. Now, distinguish between albuminous and non-albuminous seed. This is another 2 marker. Why are apple and cashew known as false fruit? Now, obviously, when you talk about the false fruit, you will get to understand why it is known as false fruit and what is the difference between false and a true fruit. Won't you know that? Right? You are not going to study the exact question out there. And if you are planning to do so, please understand, with the limited time that you are having, if you study about false fruit, please know that how it is different from the true fruit as well. And while you invest a little 5 minutes of your time to that, you probably would be able to answer some other question associated with it. Got it? The next one is write the significance of spore pollen. Very, very important thing. Okay, they can ask you in another format, why pollen grains can be stored as fossils. Why? Because they have spore pollen. What's the importance of spore pollen? Talk about it. So that's the exact question, right? The significance of spore pollen. They just have changed the question. And now give reason. Micropyle remains as a small pore in the seed coat of the seed. Why do we have micropyle as a small pore in the seed coat? For exchange of water as well as gases for respiration. You know, seed is waiting for germination process to happen. For that, you need some space such as water could get in. Otherwise, germination is not going to happen. So that's why you have micropyle. And then you have the next question, that is differentiate between pericarp and perisperm. Pericarp is the outer wall of the ovary, perisperm is the Burstensson nucleus. Talk more about it. For 2 marker questions, 2 points, nicely write it, you will get your marks. Difference between gytonogamy and xenogamy, both of them are cross pollination, but one brings about genetic variation, one does not. Okay, so talk about all of it, and it is going to be sorted. Now listen to me carefully. If any of these questions make you feel that I have not done my preparation properly, as I said, don't be worried. I'm coming in the marathon session where I am going to talk about all these questions in depth such that you do not have any issues. Any questions that are going to come from the concepts that I'm going to teach you, I'm pretty sure every one of you will be able to answer it. That's my promise. Okay, done? Deal. Fine. Now the 3 marker questions that we have are mostly diagram based questions. Okay, in your midterm or probably your boards, you can expect a 3 marker diagram based question. In FA1 things could change, but anyway, I'll show you the important diagram based question. I'll show you a question that came the previous year in 3 marker session which was not diagram based as well. So the question was, why are some seeds referred to as apomictic seeds? So basically you should know what is apomixis. Mention its advantage to a farmer who uses them. Then you have been asked about the advantages of the apomictic seeds. So if you know apomixis, you will be able to tell the advantage. If you do not understand what is apomixis, the second part of the question just goes off. Okay, so we need to know the concept. That is important. Now here, diagram based questions that have more chances to come. Draw a neatly labeled diagram of a typical anatropous ovule. That means inverted ovule. It looks like this. On page number 9, you have the picture of anatropous ovule. Go check it out. Next one, draw LS of a grass embryo diagram. What is grass embryo, ma'am? We have studied about dicot and monocot. I do not know what is grass. Baba, grass is monocot only. So you have been asked about what? You have been asked about diagram of monocot embryo. How does it look? This is how it looks. Okay, page number 19, you have this particular picture. You have to draw this and practice this. Now this is kind of complicated, which is why I'm telling you. I have done a video on one shot for sexual reproduction in flowering plants. It's a long video. I do agree to that. But one shot is done, KCET question is done, PU2 questions, everything has been done, diagrams are being done. So if you wish, you can go and watch it if you're probably watching this video today. And if you feel that, okay, ma'am's, marathon is going to start from basically tomorrow, 29th and 31st, the marathon is there. Before that, if I want to cover the one shot, I can. Please go ahead and do so. Okay, there are 2 chapters, one shot that I've done already, sexual reproduction, flowering plants, and human reproduction. If you wish, you can check it out. Now, by the way, here is a diagram of the pollen grain, page number 7 in your NCERT, you would be having it. Now, draw a neatly labeled diagram of the monocot seed and draw a diagram of the dicot embryo. So, if you ask me, this particular diagram, important, this particular diagram, important, this particular diagram, important, mostly the diagram of the monocot seed. So, monocot embryo you draw, monocot seed is different. I'll show you the picture. How does it look? So it looks like this. This is the monocot seed. Page number 21. Diagram based question might not come. You might be asked to label it. Labeling based question could come. What is this? So you should know it's coleoptile, coleorhiza and everything. And if this picture made you go nuts, let me tell you again, marathon could help you. Now, typical dicot typical dicot embryo, the diagram is going to be this. It's an important diagram, just like monocot embryo. This is also important. Now, structure of megasporangium. That means you're talking about structure of ovule. Baba, you made anatropous ovule. Anatropous ovule is nothing but ulta ovule. Here you have placenta and funicle. You just made it here and it becomes anatropous. So megasporangium structure, anatropous ovule structure, almost same. Even though the picture looks kind of little different, but it's the same. Page number 9, you can find it. Now, mature embryo sac. The female gametophyte is also known as the embryo sac. This is how it looks. You know, antibodal cells at the top. This picture could be important, so I'm just putting it as a star mark. Now, transverse section of anther. Again, what are the layers that we have? Epidermis, endothecium, and middle layer tapetum. Now, here I do not expect a diagram based question. You can get an MCQ based on what are the layers which are there. That is why I put this particular picture. So there are some pictures that are going to come in the form of labeling or in the form of MCQs. Just know that there are pictures that can come in the form of diagrams. This, and this have chances for that. Okay, cool. Yes. Now I'm just going to go ahead, insect pollination. They can give you this picture and say what type of pollination is happening over here, give the features of that. So you should know insect pollination is happening over here and talk about its feature. Clistogamous and chasmogamous flowers, you should know their features. They can give you this picture and say what are these flowers at the top, what are these flowers over here, how are they different. So please know the ones towards the soil is clistogamous, the closed ones, and the ones above will be the open flowers, chasmogamous. Again, there are 2 pictures, page number 9, important pictures. These pictures are there. MCQ based questions could be coming in. You have syncarpous ovary where there are multiple carpels are there. They are all fused together to have a single ovary. This is syncarpous. Example is papaver. Then you have apocarpus where multiple carpels are there but they are not fused together. They are different. That is Michelia. So these two examples could be helpful. So either there could be a thing you do not study this. Okay. And basically, if you see this and are getting panicked, then just know about this. Go study about it. You know that it's important. And if at all you invest a little bit of time, probably you might be getting the marks as well. There is no point of getting worried about things, Baba. Let me tell you, if you're aware that something is going to come and that worries you, understand, okay, at least I know that right now and probably I can invest my time onto it. I can probably study, right? Yes, that's the positive way of thinking that we should be having. Now, one more very important thing. So, we had launched Pariksha Veera course, which is there for KCET as well as second PUC. If you are someone who have not enrolled into it, go check it out, because this is where your entire second PUC plus KCET is going to be covered. And by November 6th, the first week of November itself, second PUC is going to be covered. And just think, imagine the situation. If your second PUC is covered by that date, you will have enough time for any preparation, whether it's JEE or probably you want to cover your first PUC for KCET. Everything becomes more feasible. And these are live sessions which are happening every single day in the Vedantu application, and there are going to be 20 plus tests. From 2nd of August, the tests are going to start for everyone. The pricing for this is rupees 3,499. The link for this is given in the description section of this particular video. Check it out if at all you wish to. Cool. Now coming back to the 5-marker questions. Now over here I have put something in yellow, 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 yellow. What is it? Basically, there could be question coming from double fertilization. There could be question coming from non-albuminous and albuminous seeds. There could be question coming from parthenocarpy and polyembryony. There could be question coming from the male and the female gametophyte, as in the pollen grain, as well as the embryo sac structure. There could be questions coming from outbreeding devices. There could be question coming from Megasporogenesis and Microsporogenesis. There could be question coming from artificial hybridization. Now when I say there could be question coming from, there could be anything. Now, what type of questions should you expect? That is also important. If I know the concept, ma'am, but I do not know what type of questions are going to come, it is going to worry me later on. So over here, because you will feel that I have studied this, but still I am not able to find the answer for this. You get it? So double fertilization based questions can come like this also. Is the double fertilization is a unique feature of angiosperms, and the products of this double fertilization is zygote and the primary endosperm nucleus. You get zygote and the primary endosperm nucleus. In the context of this, when a hexaploid plant is pollinated by a tetraploid plant, find out the ploidy of zygote and the PEN. So when these two types are coming together, what is the ploidy going to be of the zygote and the primary endosperm nucleus? You can get this type of complicated questions as well. That's the thing that I want to tell you. And for those of you who are like, oh, mama, what will I do? In the one shot, I have given you the answer for this question. Okay. Apart from it, in the marathon also, I'm going to give you the answer for this. The reason I'm not talking about that over here is that it could take a lot of time. Yeah. The next thing, double fertilization is reported in plants of both castor and the groundnut. So if you know, castor is a dicot and groundnut is also a dicot. However, the mature seed of groundnut is non-albuminous and the castor is albuminous. Non-albuminous and albuminous. Explain the post-fertilization event responsible for this. So if you know that albuminous means endosperm is persistent, non-albuminous means endosperm is not persistent, you will be giving the answer like that. That basically after fertilization, you have been asked about post-fertilization. After fertilization, usually the endosperm is being formed and it is being used up. But in some cases, the endosperm persists. Just like that, those are albuminous. An example for that is castor. Okay, similarly over here, you have the endosperm sometimes does not persist. It is used up in the formation of cotyledons. Okay, and this is usually the case of dicots. For example, groundnut. Groundnut is a dicot that is having non-albuminous seed. It does not have any endosperm persisting. Rather, it makes the cotyledon out of it. So, when you know these terms, you will be able to answer it. You will be able to give more points around it. That's what I wanted to say. Next thing, banana is parthenocarpic fruit, whereas oranges show polyembryony. How are they different from each other? Now, if you know what is parthenocarpy, you know that there is no seed. There is no seed formation that is going to be there. That is how it is going to be different. In polyembryony, in one particular seed, you will be having so many embryos. Get it? So that is a different small question. You can get answer. A and B type of questions have come. Both of them are small terms. Still, you can get you 5 marks. Now, explain the number of cells, number of gametes, ploidy of cells and type of cell division involved in the formation of male and female gametophyte. So, basically, embryo sac, how is it formed? What is the ploidy of all the cells? Similarly, pollen grain, that is a male gametophyte, how is it formed? Again, ploidy of all the cells which are there. There are 2 cells, vegetative and generative. Generative divides again, forms, the gametes are there, know their ploidy. Important. Okay, next thing, explain the different outbreeding devices. Straight question. Different outbreeding, or they can twist the question and give you, if at all the plant does not want to do self-pollination, what are the steps that could be used by the plants? So basically you have been asked about outbreeding devices. Next, which advantage offered by the seed help angiosperm to adapt to changing environmental condition? That means you have been asked about the importance of seed out there, the advantages of seed. Now arrange the cells in the order of microsporogenesis, megasporogenesis. When you are given cells and you have been asked to arrange them in the order, you should know exactly the flow of this and this. Now while you study this, will you be able to answer this question? Yes. Got it? First MMC, then a device, then a device. That is how you get it. Now, this question is very interesting. Plan an experiment and prepare a flowchart of the steps that you would follow to ensure that the seeds are formed only from the desired set of pollen grains. Only from the desired set of pollen grains. That means you're talking about artificial hybridization or controlled pollination. Now, they have not mentioned anything like that, but you have to understand that this question is from that topic. Got it? And then finally, water and insect pollination, the features of the plants which are there in water as well as insect. Also, wind is finally important, but please make sure you know their features really well. The points, there are four points for each of them. Just know about it. Again, if you feel that you study the concepts that I have marked in yellow, if you're not able to solve these questions, do not worry, I am coming for the marathon session. And before I end this particular video, I want to ask you, when do you have your FA1 examination at your place? Please let me know in the comment section, and do let me know what's the next chapter that you want. And do not forget to hit the subscribe button for getting all the videos like this. One more very important thing for children who are like, ma'am, I do not understand, where do I find everything? Because whatever is happening in YouTube, I'm not able to find everything at one place. So under every video, you will be able to see this free ACE KCET 2027 course. Click on this. This is actually an all-in-one page. Okay, you can just move down, click on buy now. It's a free course. That's why it's for zero. Everything which is there, you just have to put your mobile number and you will be able to access it. Every academic session which is there in YouTube will be done separately, will be posted up over here. It will be visible for you. Go check it out. Good. So that's all. Wishing you the best. Please take care. Bye-bye. Lots of love.